Hello, everyone. My name is Helia Rahimian, and I'm an undergraduate researcher for the Center for Tissue Engineering. Our lab central focus is on adipose-derived stem cells and their use in wound healing solutions, scar control, and cartilage framework development. In this project, we looked at enhancing fat graft survival through adipose-derived stem cell-induced vascularization. Autologous fat grafting is one of the most popular procedures in plastic and reconstructive surgery. The surgeon starts with selecting a suitable area with excess fatty tissue, extracts the fat using liposuction, and refines the fat from any fluid, blood, or cell fragments before turning it into an injectable form. Unfortunately, resorption rates are very high and variable. So this turns our focus towards vascularization or blood vessel growth of the fat graft. Vascular endothelial cells are the cells that form the inner lining of blood vessels and are responsible for nutrient and oxygen exchange with neighboring tissue. When these endothelial cells fail to divide and proliferate, inadequate blood vessel formation may cause ischemia, necrosis, impaired healing, and in this case, fat resorption. Adipose-derived stem cells, or ADSCs, are multipotent stem cells that have the ability to differentiate into many cell types. ADSCs demonstrate proliferative and regenerative potential due to their secretion of cytokines and growth factors. Therefore, adding ADSCs to a fat graft benefits tissue revitalization by encouraging neighboring tissues to heal and form new blood vessels. Currently, studies suggest that endothelial cells rely on glycolysis to produce ATP. Therefore, a more glycolytic cell results in a more angiogenic one. We've proven that endothelial cells undergo glycolysis by performing a lactic acid assay. We saw that by inhibiting glycolysis, there was a reduction in lactate production, which is a byproduct of glycolysis. Additionally, we established that ADSCs have an autocrine ability to secrete glucose, and endothelial cells take up this glucose in order to produce their energy. So we hypothesized that adipose-derived stem cells can improve fat graft retention rates by regulating glucose metabolism in endothelial cells, thereby stimulating angiogenesis and promoting vascularization. We analyzed glucose uptake in endothelial cells when cultured in media that lacks exogenous glucose, and the little glucose uptake seen was due to an autocrine secretion, but that was blocked with glucose transport inhibitors and an inhibitor that blocks glycolysis. However, when we co-cultured endothelial cells with ADSCs, we saw a significant increase in glucose uptake even when treated with inhibitors of glucose transport and glycolysis. These findings suggest that when glucose uptake and signaling pathways are blocked in endothelial cells, the presence of ADSCs overcome this inhibition. Further studies are required to elucidate whether ADSCs are initiating an alternative pathway for glucose entry into endothelial cells. Thank you for listening.